Students, now we have to discuss the various steps involved in DNA fingerprinting technique. Now we need a cell with nucleus. The RBCs cannot be used for DNA fingerprinting because of the absence of nucleus. So we need any cell, either a living cell or a dead cell for DNA fingerprinting technique, but the cell should have a nucleus because DNA is present only in the nucleus. Now the step number 1, extraction of the sample of DNA. So you have to obtain a sample of DNA either from the blood cells, that is WBCs, or from the hair cells, the root cells, hair roots. Hair roots only contain living cells. Okay, that's why you have to take hair root cells, or the skin cells, or from what we have the bones, or from the teeth. This is what is called extraction. So step number 1, extraction of DNA. This is step number 1. So it may be I am taking here the blood sample. So we can take, for example, a hair cell, the root of the hair roots. So we are extracting the DNA from these blood cells both together, obtaining the blood sample and extracting the DNA from the sample. Now the next one, the DNA fragmenting, and before that, normally you see that one, PCR reaction, PCR. So why PCR reaction is needed? Polymerase chain reaction. This is normally used for gene amplification or DNA amplification. That is increase in number of copies. Suppose the amount of DNA extracted is actually not available enough. At times it is needed to produce copies of what is called the DNA fragments through one stage what is called polymerase chain reaction. We will be studying this one under chapter Applications of Biotechnology. Now in the case of polymerase chain reaction, we have produced millions of copies from a single segment of DNA. This is called gene amplification. So the second step is nothing but polymerization reaction, PCR reaction, polymerization chain reaction. Now we are getting many. Now the next one, what is called digestion, the third step. So the second step, this is second step. Though I am mentioning here and there, but normally we have as per the book paragraphs, extraction of what is called the DNA. The number 2, polymerization. So what we have polymerization chain reaction. Polymerization chain reaction. The next one, digestion of the DNA. The meaning for that one, that's called the fragmenting of DNA. So this is the one, fragmenting of DNA. What is the meaning for that one? The entire DNA is normally treated with restriction endonuclease enzyme. The restriction endonuclease enzyme cut the DNA into fragments of different sizes and specific size. This is what's called fragmenting of DNA, otherwise we can say digestion. So in fragmenting of DNA, the DNA is treated with the restriction endonuclease enzyme. That enzyme breaks the DNA into fragments of specific size. This is what we have. The next one, just actually separation of the DNA fragments through electrophoresis. Separation of DNA fragments through electrophoresis. Now in the case of electrophoresis, the main idea is that as the DNA is negatively charged molecule, it sees through the agarose gel plate. If you are taking agarose gel plate, now this is what we call the gel plate. It is having many wells. This is what's called the well. Here the DNA fragments are added. Now this gel plate made up of agarose gel, we will talk about under biotechnology, agarose gel. Now this agarose gel plate is placed in a salt solution and it is being connected with positive and negative charges. Now the DNA fragments are actually of different sizes. They are moving through or just receiving through the agarose gel towards what is called the positive electrode as they are negatively charged. So the fragments which are smaller in size, that is normally just moved farther apart from the well. This is called as a well. The pocket-like structures, the cavities, they are called as well. When fragments are placed, They are passing through the agarose gel, sieving through the agarose gel, or diffusing through the agarose gel according to the size. So we have to separate the DNA fragments into different sizes. Now when we have supply electricity, now as the DNA is the negatively charged molecule, it moves towards what is called the positive electron. While moving through the agarose gel, it produces what we have bands, separated bands. Now we are getting separated bands. So this is what is called the agarose gel plate. Having cavities, these cavities are called what is called the well. We are placing the fragments here. This is being placed in a solution, isotonic solution, connected with positive and then negative electrodes. Now the DNA is moving, and the DNA fragments are moving as they are passing through the what is called the gel. What is happening? We have received separated bands of different sizes. Separate bands of different sizes. For example, this is a band. What we are getting in the form of separated bands. Bands of different sizes. Some fragments reach actually towards the end, farther apart from the what is called the well. They are considered as what we call the smaller fragments. So the size of the DNA fragments are represented in the form of what is called separated bands of different sizes. The one which moved farther apart from the well is the smallest. The one which is found nearer to what is called the well is the largest fragment. So during electrophoresis, as in agarose gel, so we have formed the DNA fragments form separated bands of different sizes. They form separated bands of different sizes. Now these bands are actually the next step normally we have. These bands are transferred or sieved out into a nylon membrane. Not now, this is actually what's happening. Or nitrocellulose membrane. So these bands are transferred normally. So that's why I mentioned the word, they are sieved out. They are sieved out from the gel plate to the nylon membrane. Now the nylon membrane is treated with what we call chemicals that allows the nylon membrane to break the hydrogen bonds of the DNA. So they are single-stranded. So the main purpose of actually what we call separation of the fragments through electrophoresis, it is a method adapted to separate what is called the fragments of different sizes. Not to sequence actually the DNA fragments, but to separate the bands of, separate bands of DNA fragments. So we are getting only in the form of bands only. And these bands are sieved out, transferred to a nylon membrane. We will discuss in the next step. And these nylon membranes are treated with the chemicals. These chemicals allow this nylon membrane to break the hydrogen bonds of the DNA molecule, that is between the two strands, so that the DNA strands are single-stranded. This is what we have actually the third step. So number one, actually obtaining the sample. Number two, the PCR. Number three, what is called DNA fragmenting. Now this is the fourth one, not the third one, the fourth one. Now we have to denature the DNA from the gel. Denature, that is removing from the gel using some alkaline chemicals or by heating. This is not needed now. Normally, if you want to denature the DNA, they are being treated with alkaline chemicals of this what is called the gel. Along with, you see, either gel can be treated with chemicals or by heating we can denature the DNA. Now what is the next step? We have the gel plate. The gel plate is having actually bands, separated bands of different sizes. The next one is called what is known as southern blotting. The next one is called southern blotting. What is southern blotting? Now we are taking what is called actually, this is a chamber, a strand, a single or small fragment that is what is called small fraction DNA strand, or we can say size fraction DNA strand. There is a strand. Now in this one we are taking what is called actually that is salt solution. Salt solution. I am telling about what is called blotting. Blotting is nothing but simply transferring anything from one to another. Even in the case of, you see that when if you have excess of ink, you are using the chalk piece or what is called blotting paper. So the ink is transferred to the blotting paper onto the chalk. That's what is called the blotting. So we are taking the salt solution. In the salt solution we have to place what is called a sponge. This is a sponge. This is a sponge. And over the sponge we have to place this gel plate with the bands, separate bands. Now this is what is called the gel plate. So here we have the separate DNA bands. This is what is called actually the gel plate. So over this gel plate we have to place a membrane. I mentioned already the membrane placed over it is nothing but the nylon membrane or nitrocellulose membrane. So we are placing over this one. The green one represents what is called the third layer. First layer from bottom you have sponge. Next layer we have the gel plate. All which we have to place what is called a membrane, nylon membrane. This is nylon membrane. So why we are keeping this nylon membrane and nitrocellulose membrane? To transfer these bands into this membrane. Then over it just to remove the excess, we have some towel over it. So I am taking this one, some towel. So this is a towel. To remove excess of what is called the probe. So this is what is called actually the blotting. So what is blotting? Normally what is happening during the process of blotting? So we are taking what is called the gel plate. Now we have size fractioned DNA strand. Now it contains what is called salt solution. Now the layers are first sponge, that is number one. Over the sponge we have to place the gel plate with what is called the separated bands of that is DNA fragments. The third one over which we have to place a nylon membrane in order to transfer the separated DNA bands to the membrane from the gel plate. And over this one we have to place a paper towel. This is the one. To remove excess of that is what is called the probe. Now what is happening? What is blotting? So it is nothing but the transfer of what we have separated DNA bands from the gel plate to the nylon membrane. So it is transferred, when placed over it. Simply just we are making a Xerox copy. We are making a Xerox copy. So the bands are simply transferred to the nylon membrane. And that process is called blotting. And this is a method what is called southern blotting. The technique is called southern blotting technique. A method of transferring what is called the separated DNA bands from the gel plate to the nylon membrane. This process is called what is known as blotting. The technique is called southern blotting technique. See that one? The DNA band pattern is transferred to a nylon membrane by a technique known as southern blotting. So this is we have transferred what we have. That is the DNA fragments or the bands here, not the DNA fragments, the bands from the gel plate of different sizes to the nylon membrane. The next step is using radioactive probe. Probe is the word used for anyone who inquire. So using radioactive probe to identify specific DNA. What type of DNA, whether it is matching with one person with another or not. Now, what is a probe? Radioactive probe. It is nothing but DNA labeled with radioactive substance. The DNA labeled with radioactive substance is called probe. You have to prepare what's called the radioactive DNA probe. So, we have to incorporate the radioactive isotope into your DNA and that is called DNA labeled with radioactive substance, and that is called as a probe, inquiry. Now the next step, what's happening? Now this DNA probe attaches the separated bands by base pairing technique, by base pairing with those fractions which are complementary to its sequences. So the base pairing occurs between the bands. So this is nothing but a DNA sequence, and what we call the radio-labeled DNA. It is having its own base, actually bases. So base pairing occurs only between what is called the bands of this nylon membrane and the radio-labeled DNA having what we have a film. So this radio-labeled DNA matches or pairs with what is called only specific fragments of DNA by base pairing process based on the complementarity, and those sequences which are complementary to its sequence. Suppose we have, this is a radio-labeled DNA having the sequences, I am writing simply, this is the sequence. And now here we have the sequence also present in this nylon membrane in the form of separated DNA bands. That is what we have. So here what is the process? Now we have the bases in the form of TT, AA and then GG. So this radio-labeled DNA forms a base pairing based on the complementarity. So with those actually the bands having the sequence which are complementary to its sequence. So by which one can find out, that is which segment of DNA is normally complementary, which is normally just similar to two different persons. So the purpose of this one, the radio-labeled DNA attaches by base pairing to those fractions of DNA based on the complementarity, and those fractions of DNA having the base sequences which are complementary to its sequence. Okay, this is what's called radio-labeled, that is method using radio-labeled DNA to find out which is forming a base pair based on the complementarity. So only certain bands, separated fragments can form a pair with what is called the radio-labeled DNA fragments, not all, by which we can find out whether it's matchable or not, whether the suspect is a crime or not. Okay, now, so this is one. So instead of using also the radioactive substance, we can also use fluorescent substance or radioactive isotopes for, that is identifying, that is a DNA segment, and that is used as a probe. So the probe can also be produced from fluorescent substance and also from the radioactive what is called isotopes. Now the pairing between what we have the bases in the nylon membrane, that is the DNA fragments, the bands, and what we have actually the radio-labeled DNA, this process is called hybridization. The pairing between the base sequences which are radio-labeled with what we have the base sequences formed in the bands of nylon membrane, this process is called hybridization. That is pairing between complementary base pairs of these two. This process is called hybridization. The next one, hybridization once again. After hybridize, so we have to remove excess of what is called just the probe. The excess of probe is washed out. Then a photographic film is placed over what is called the membrane. So we have the first one, radio-labeled one. So this is nylon membrane, over which we have what is called this radio-labeled DNA, and over which we have to place a what we have a photographic film. That's nothing but the X-ray film. So which is normally having hybrid DNAs. Hybrid DNA is nothing but the base paired DNAs. So this is what's called base pairing. These DNAs are called hybrid DNAs because they form a base pair. That process called hybridization. The resultant DNA is called hybrids DNA. So we are placing, so first one nylon membrane, over it we are placing a radio-labeled DNA. The third one we are placing a photographic film over what is called the membrane, the one which has hybrid DNA. So what's happening? The next one, exposure on the film to make a fingerprint. So now we are placing one nylon membrane, the next one was the radial DNA, the third one we are placing a photographic film, nothing but the X-ray film. The last stage is nothing but the exposure exposure on what we have the photographic film to make a fingerprint. Now the radial labeled DNA exposes to the photographic film or the X-ray film to form images. So now the images, because of radioactivity, now it is being formed an image that is in the film. So image is formed. So images of bands are produced in what is called the photographic film. So we have transferred from the gel to the nylon membrane, then pairing of the nylon membrane bands with what is called radial label, what is called just we have the DNA. Now we are placing a photographic film over it so that we can get the images of what is called the bands. These images, the bands of images are corresponding to specific DNA fragments. Now we have received thick and thin dark bands. They form what is called bars, patterns of bars. You have bars, this is what we call the bars, patterns of bars. These bars are, we have the bars like this. So these bars represent what we call the DNA fingerprint. So on the radial label actually DNA exposes to the film, images are formed, images of bands are formed. These images of bands in the film corresponding to the specific DNA bands, and we have received thick and thin dark bands. They form pattern of bars. These bars represent the genetic or what we call fingerprint. So that's about the various steps. I represented here, but the things what I described in a different manner. Now we see that one excess DNA probe is washed out, that is after hybridization. Now at this stage the radioactivity probe is bound to the DNA pattern on the membrane. So binding, that is a pairing. That is called hybridization process. The pairing between the bands of nylon membrane and the radial labeled ones of DNA is called hybridization. So X-ray film is placed next to the membrane to detect the radioactive pattern. So we are just actually see that one. We are placing a film to get a photograph, just like what we are taking the image of any part of the body by using X-ray. The same manner, placing a photographic film or X-ray film. As it is radioactive, it is being normally just X-ray to get images of bands. These bands are corresponding to the DNA bands. They represent what's called the DNA fingerprint, or that is we can say genetic fingerprint. Now what are the uses of DNA fingerprinting? So it is used in forensic analysis. You know the forensic studies related to just the determination of the crimes, the criminals, and also just finding out the disputes between two parents while claiming just a single child. Now number one, that is forensic analysis. Now there are three aspects here. One, identification of those persons who are involved in criminal activities. That is number one. Number two, there are always disputes, maternity dispute and paternity dispute. A single child is being claimed by two parents. In such cases, to settle the maternity and paternity disputes we are having, there is what's called DNA fingerprinting. The DNA fingerprinting is taken for two different people. Those are claiming a single child, and by matching pattern we can conclude only one parent is for that child. This is called maternity and paternity disputes that can be solved through this forensic analysis of DNA fingerprinting. Another one, determining a relationship for immigration purposes. Suppose a person, that is the parents settled in India long ago, but their children went to Pakistan. They wanted to enter India. In such cases, whether is there any relationship between the parents in India and those are immigrating from Pakistan, those who wanted to settle in India, they are claiming that their parents are here. In such cases, they are making a DNA fingerprinting between the parents and the children, those are immigrating from other countries. And that purpose also we are using what is called, that is the DNA fingerprinting. So that is about forensic analysis, identification of persons involved in criminal activities, settling maternity and paternity disputes, and also determining relationship for those immigrating into a particular country. Now the pedigree analysis. So the pattern of inheritance, the course of inheritance of genes through what is called just generations. That is the pedigree chart we are making, the pattern of inheritance of genes through generations. And also to find out the course of genetic disorders from the parents or the great parents to the present child, how far the character is being inherited. That is for detecting genetic diseases. Now the third one, conservation of wildlife. That is nothing but the protection of those individuals which are at the verge of extinction. They are called endangered species. By maintaining the records of DNA for the determination of the tissues of such individuals, now we can prevent the dead endangered species. So we can identify the dead endangered species by means of what is called the DNA fingerprinting, because of maintaining the DNA records for the identification of the tissues of dead endangered what is called the species. Now the last one, anthropological studies. The study of humans is called anthropology. Now here, determination of origin and migration of humans, where the first man originated, that one in African countries. Then how did they migrate to different parts of the world? That one can be found only with the help of DNA fingerprinting. And also, we can find out the genetic diversities among the human race. The genetic diversity, differences in their genetic nature, genetic diversity, different kinds of races, Australian, Mongoloid, different kinds of races we can just find out, that is called the genetic diversities in these human races. So these are some of the uses of DNA fingerprinting. So with this I concluded the DNA fingerprinting, just also the lesson molecular basis of inheritance, the longest lesson in your syllabus. And go through thoroughly, then only you can understand. That is the materials what given in the book and also what I taught you. There are some actually mistakes in the book. I rectified those mistakes, I didn't mention those mistakes, even for transformation. And this transformation, the definition is given wrongly in the book. So I mentioned already what is the correct definition and follow it. Okay, thank you.